speaker will be Dr. Anita Krishnan from Biocon. So, Dr. Anita Krishnan comes with a wealth of experience and she represents the industry today in this forum. 19 years of experience in research and development and 14 years of leadership in biosimilar product development. She is experienced in both physiochemical, structural, functional characterization of biopharmaceuticals, monoclonal antibodies, fusion proteins, therapeutic peptides, and so on. She is a CMC expert from end-to-end biosimilar product development for global markets, providing direction to cross-functional process and analytical teams. She is well versed in life cycle management of methods. She is also an expert panel member at the United States Pharmacopoeia of Multi-Attribute Methods, Mass Spectrometry. She is also an alumni of the Indian cohort of women leaders for the leadership journey with Women Lift Health. Thank you, Anita, for joining us today.
First of all, a thank you to the organizers for inviting me. It gives a chance to give back to the society. As a woman scientist, you go through a lot of trials and tribulations. It's not easy to reach where you are, all the presenters who have presented. So I'm happy to give back to the community. This is a slide on the company that I represent. I work for Biocon. I'm the head of analytical sciences in Biocon R&D. Biocon is a fully integrated biosimilars company. Biocon is just down the line. I'm not sure how many of you know. We have 3 campuses here, one in the Jigni Road and one in Hosur Road, which are both our manufacturing plants. And then the corporate office is on the other side of Hosur Road. So we actually have 3 campuses here. So these are the 3 major divisions in a biopharmaceutical company. This is R&D, manufacturing and commercialization. I represent R&D. In R&D, we have capabilities for process and product development. And then we have capabilities for scale-up and tech transfer to manufacturing plants. We have all capabilities for analytical and bioanalytical. Analytical means it's physicochemical and structural and functional characterization of the protein therapeutics. Bioanalytical sciences means that when you inject the sample or the therapeutic into the patients, then you have to look at the PK, PD and safety and efficacy. So that is bioanalytical sciences. We develop and transfer the methods to GCP labs. Clinical development, our molecules go through clinical trials and hence we have a separate section taking care of clinical. We have physicians, we have medical writing teams and clinical operations who conduct the clinical trials across multiple geographies in the world. Intellectual property, obviously, because we are in the industrial research, we have a division for taking care of our intellectual property. Manufacturing, we are one of the few companies which have all the expression platforms. We have capabilities for mammalian and microbial technological platforms. They are very different in terms of physiology and cost and design. Insulin, we also have a wide variety of portfolio molecules, which is insulins, monoclonal antibodies, and conjugated proteins. We have capabilities for drug substance, that's just the drug itself, drug product, which means it is a formulated drug, and also the device. So if you have an injection or an auto-injector pen, which you pierce yourself multiple doses, that's a device. So we have capabilities for devices also. We have 3 manufacturing sites. As I said, 2 sites are just here, one on the right, one on to my left, and we have one more site in Malaysia, which is the major site for production of insulins and insulin analogs. All our manufacturing facilities are CGMP. CGMP approvals are nothing but current good manufacturing practices. So we get approvals from the regulatory agencies. We have approvals for more than about 85 countries. Commercialization was not part of our company. We were collaborating, so it was partner-led earlier, but recently we have acquired Viatris and hence now commercialization is also part of our company. A commercialization, basically it is advanced markets as well as emerging markets. Earlier we were only doing it in emerging markets. Now we also are able to commercialize in advanced markets. And we have 8 products, 8 biosimilar products commercialized globally. I'll talk about it in my next slide. And we still have a self-led and a partner-led model. We have both models existent in the company. Next slide, please. For a person in R&D, this is like looking at your paper published in a high impact journal, because as a R&D scientist, you don't get to see this. Like the molecule that I'm working on currently will get launched in 2030. So to look at a final product which is launched in the market and which really helps patients is a big boost to any R&D scientist. So these are the biosimilars that are approved in oncology segment. We have pegfilgrastim, which is used in cancer care, which is after your chemotherapy, you need this. So that's for cancer care. Then you have trastuzumab and bevacizumab. Trastuzumab this is world's first monoclonal antibody approval from US FDA, which was given to Biocon. This is bevacizumab, which is a product for lung cancer, which is Abvemi. In the diabetes section, we have insulins and insulin analogs. Those are insulins, and these are modifications of insulins for longevity in the patient. This is recombinant insulin, aspart and glargine. On the immunology side, we have adalimumab and etanercept. These are autoimmune diseases, drugs indicated for autoimmune diseases. You see the number over there because they are partner-led, and now they have been, because we have acquired the company, it is now part of Biocon's portfolio. Incidentally, I was the scientific lead for etanercept in my earlier company. So it's like a baby joining back the family kind of a feeling. So it's now part of our portfolio. Next slide, please. Next slide, please. Yeah, so now to the science part. What are biologics? We just heard a lovely presentation from Dr. Mrs. Balram. Biologics are protein molecules. If you see onto my right, and onto the left is a small molecule. I just put this up to show you what is the difference in the size between a small molecule and a large molecule. These are the sizes. You have 180 Daltons, you have 5,000, 22,000 and 150,000 Daltons. So the more bigger the molecule, more is the complexity and more is the requirements to show similarity. And the complexity also arises because they can only be produced in living systems. They cannot be chemically synthesized. So when they are chemically synthesized, it's easy to show their purity and then just inject into patients. Whereas once they are very big in size, then there are multiple post-translational modifications that come into place. Okay, before we go to, so I just wanted to run through a little bit on the regulatory pathway. As I said, for a basic research scientist, you feel accomplished when you publish in a journal. It means your research work has been accepted by the community. For a manufacturing scientist, for a person who works for the industry, if your dossier gets accepted by a regulatory body, regulatory agency, that's a sense of fulfillment. For that, these are the different pathways. I've just taken US as a case study here. What you see on the left is a pharmaceutical molecule, small molecule. This is small molecule which is new, and this is generic, a copy of the small molecule. Small molecule is nothing but like you eat your Dolo, right? That's a small molecule. It's chemically synthesized solid orals. So that goes through a pathway called 505, B1 and B2. It needs full report of safety and efficacy. It needs full clinical trials, which means there are about 4 different clinical trials that are needed. Whereas if it is a generic, once the patent is over, you make copycat versions of that. You still don't have access to the process what the innovator has done. You re-engineer the whole thing yourself, but since it is identical to the small molecule, you do not need to do any clinical trials. Similar is the case in biologics. In biologics, it goes through a 351A and 351K pathway. The innovator has full report of safety and efficacy. Since it is a biologic molecule, I just spoke about the complexity of the molecule, it still has to show highly similar, and some level of clinical trials is still needed for the 351K pathway, which is biosimilars. Hence, the biosimilars are not as affordable as a generic molecule. Next slide, please. Okay. So this shows an inverted pyramid here, which shows what is the level of regulatory emphasis on both these pathways. Now I'll talk only about 351A and 351K pathway. 351A is what you see here, which are new molecules, which is basically a discovery molecule, which goes through full clinicals. So the dark blue is very low regulatory emphasis. That means the regulatory lens or the scrutiny is lower as compared to the light blue. So in the innovator pathway, molecular characterization, yes, needed. Non-clinical studies, yes, needed. Phase 1 clinical studies needed, but the regulatory emphasis till here is lower. When it comes to the phase 2 and the phase 3 clinical trials, this is of high regulatory emphasis because this leads to the claim of a new drug for the innovator. Hence the emphasis over here is very high. It's exactly opposite for a 351K pathway, which is biosimilar. Here you are making a similar molecule. So the molecule itself is not different, but it cannot be called identical. Hence, physicochemical and functional characterization is of very high requirement. And as you go higher, you see that it is now dark blue in color. It means it is just a confirmatory clinical trial that you do. You are not discovering anything new in the clinical trial. So in my talk, I will just walk you through what is a stepwise process for development of biosimilars. These are the different steps. First, you define the CQA. CQA means critical quality attributes of the drug therapeutic. And then second part is, once you define the target, you have to consistently produce the product. That's the second piece. Third is, once you produce the product, you have to demonstrate that it is highly similar structurally, physicochemically, functionally, in every way possible. Fourth is, because it is produced in living systems, invariably you will see differences. It will not be identical. Do you understand the differences? Have you studied the impact of those differences? That is the fourth step. Fifth step is again confirmatory safety and clinical trials that you do. These are the 5. I'll run you through each step. Next. Okay, so we'll take a case study of a monoclonal antibody. This is the large class of biosimilars are monoclonal antibodies, and this is how a monoclonal antibody looks. It is multi-chain protein, so proteins will have primary, secondary, tertiary and quaternary structures. And when you have subunits like this, it has all the levels of structures. That means. So it has 2 heavy chains and 2 light chains, and you see the number of amino acids that are present in each chain. And you are also able to see how many disulfide bonds are present over here. It has, an IgG1 has about 32 disulfide bonds which are bonded, and which means there are 16 disulfide bonds. So it is a heavily linked and a clear structure of a Y structure. A Y structure is made so that it can, catch on to the antigen. Basically, it binds to the antigen in the Y. It has certain complementarity determining regions, CDRs, which bind to an antigen. An antigen in this case can be an overexpressed ligand or a receptor, whatever is the indication. And each of these amino acids, several of these amino acids can go through post-translational modifications. I'll just take an example. If there is methionine, it can get oxidized. Out of 440 amino acids, let's say there are 20 methionines. 10 of them can be oxidized, and 10 of them need not be oxidized at the same level. So this level of variations you will have, these are called variants of the protein. Now your product should have similar level of those variants and same variations, and if you have different variations, you will have to justify whether it has an impact on safety, efficacy, PK, PD, all of it, right? So, which means we start our development by reverse engineering from the product. So the products that I just showed you are biosimilar products. Similarly, if we target to make a particular drug, we source it from the market and we sequence the product. We start from there, and the target range, quality range is actually determined from the product itself. Next slide. This is how it looks. So we procure the product and so we procure the product and analyze it for its quality attributes. So each of those attributes will have some range. We just spoke about methionine oxidation. It could be 2%, 2.5%, 3%. So you map it like that. Okay, and when you start, you know it is an IgG1, and so because of your prior experience with this class of molecules, you kind of know what to expect. Although several of them will be quite similar, for every product there will be something which is different. So it means you have to do a product specific molecular assessment first. understand the molecule thoroughly, and then map your quality attribute. It's something like when you want to build a house, first you make a plan, right? You want the kitchen here, you want the room here, you want windows here. So you make a plan. So QTPP is nothing but quality target product profile. This is my target for development. Okay, I want this presentation, I want a pen device, all of that. And then each of these attributes, the product quality attributes, you will rank it as high criticality, low criticality based on all of these inputs from literature, from regulatory documents, and also some internal experimental work. Once you do that, you have a list of CQAs, which is your target. CQAs means it is a critical quality attribute. It means you have to have. You have the human body. You have to have 2 eyes. You have to have one nose. You have to have a mouth. But your hair can be longer or shorter. It doesn't matter, right? So that is the criticality you will list. Next slide, please. I'll move to the next part, which is produce consistently. It's a busy slide, but I can just walk you through this. This is the whole process of production. Okay? We start with the amino acid sequence. We just spoke about the amino acid sequence. So this is protein sequence. We go behind, we go back to construct the DNA sequence, inject it into the cell. Now you have the cell producing the protein that you want it to produce. It has been inserted into the cell in a way that it has been instructed to produce this protein constantly. When you inject it into cell, you will inject it to multiple cells. Let's say it is random integration. It integrates in many places. So each clone will produce a product in a different way, in a different level, which means it will go through a clone selection process. You will screen all the clones and you select. Basically, you are looking at that one single cell which produces the maximum protein for you and the best in quality, right? So you will do the screening. Every time you do screening, I spoke about quality, you will come back to the critical quality attributes. You will be doing testing. These are all in between the process you do testing. Once you find a clone which is producing the protein, you will take it into production. This is various levels of production. You will start with the tubes and then you will go to the bioreactors. So in R&D we have a lab scale. We will do it at 25 ml, 50 ml, 1 liter, 5 liters. We have multiple scales. And that's what we do to set the process, because you will do process development to get the right quality and to ramp up the protein expression also. So that's what you do. Once you get the protein, now protein is a part of multiple other supernatant proteins, which means you have to purify the protein, the therapeutic, to a highest level of purity. That is when the chromatography kicks in. Chromatography is nothing but liquid chromatography. You have a stationary phase and a mobile phase. You use the biochemical properties of the protein to bind it and elute it at the way you want, so that the rest of the proteins wash off. So you can employ more than one chromatography operations, and again you will be testing it everywhere to see that the quality matches. And then once you have the pure protein, you will then formulate what is formulation? Now you have a protein. If you simply inject it into your skin, you will get rashes, because your skin has a physiological salt balance, it has a physiological pH, right? And the drug, once you put it into, let's say, in a pre-filled syringe, it will be lying in your pharmacy for 2 years. It has to be stable. So all of that comes under formulation, which is the closest to the patient, right? All of this is much earlier. But what goes into the PFS is closest to the patient. So there is a lot of scrutiny over there, what kind of water you use to make those buffers and things like that. So the level of scrutiny over there is very high. You see a red star over there, that is endpoint testing. So if you do testing only there, you will have more rejections, right? So that is why we do testing all over. We call it in-process testing. Today, manufacturing wants quality to be ingrained in manufacturing, which is quality by design. So unless you do testing in all these places, you will not have control of the process. For example, if you see the top there, there are glycan variants. There are sugar molecules which are attached to the monoclonal antibody. Now sugar molecules cannot be changed in chromatography. It is a clonal property. It means while doing the clone selection itself, you have to select the right clone, because post that you will not be able to modulate the glycan. To some extent you can modulate it in upstream process. Beyond that you will not be able to modulate. Charge variants. Charge variants can be formed in upstream, can be controlled in upstream, could be formed or may be formed because of hold time in downstream. and during stability they can increase. So once you know this, once you have this understanding, you will know how to control it through the process. And that know-how in the lab scale has to go to the manufacturing scale, and they have to reproduce it exactly the same way. So which is why I spoke about continuous manufacturing and automation, because in R&D you have very skilled, very brilliant scientists who know everything. But once it goes to manufacturing, it's a set of instructions that need to be followed to the T, exactly to the T. Now when there are modulations over there, the quality goes for a fix, and then you are not able to produce the product consistently. That's the catch. These are the different scales that I have written. Lab scale is generally used for development. Pilot scale. So what we do is, towards the end of lab scale, we come to the pilot scale, which is normally used for animal studies if it is needed. But pilot scale is the proof of concept that you can take it up as a scale up, so that you can go to the clinical scale. Next slide. Okay, so this is the third part of the talk, which is demonstrate similarity. Once your protein is produced, whether lab scale or whether it is your pilot scale or whether it is your other commercial scales, you need to test the molecule, that is protein molecule, using several techniques, looking at the primary, come to here. There are several product related substances and impurities. Why we call it substance and impurity? If it is just a variant but has no impact on function, we call it substance. It means it can still be present. But if it's an impurity, it means it has an impact on the potency. It cannot be present and needs to be controlled. So you have primary structure, higher order structure, variants, impurities. You also need to show the degradation kinetics. So over a period of time, 2 years or 3 years, there will be some degradation of the product, but yet till the end of the shelf life, it still needs to be potent. So that also needs to be experimentally verified and data has to be generated. So all of this comes under, so you have multiple techniques over there which are looking at glycans, proteins and charge variants and size variants, clips, fragments, high molecular weight impurities. Each of it has a different impact on the safety and efficacy of the patient. All of this needs to be done on that product. So for a single product, we run close to about 40 to 45 different methods, orthogonal techniques, to look at it in so many different ways to document its purity and potency. Next slide, please. So what I showed you was structural and physicochemical. This is functional. Protein structurally, most of the techniques could be similar, but functionally the protein can be very different. So depending on the function, you have to also design your assays. I've just picked up is an anti-TNF molecule. So you will have as a molecule the Y-shaped structure. The Fab portion, that is the anterior portion, binds to the antigen, and the posterior position, that is your stalk of the Y, it attracts other cells, because the job of a IgG is basically to catch hold of a virus and kill it, right? That's the primary role of an IgG. So the antigen binds here, and the Fc portion attracts your soldier cells, all your immunological cells which come bind, and then it brings it, the proximity kills that virus. That's the purpose of the IgG. So in this case also, it will attract some of your immunological cells, which is PBMCs, NK cells and things like that, which will then kill the target cell. Target cell could be a cancer cell in this case. So you will have to show all functions of the Fab, all functions of the Fc, and most of these assays will be run in comparison with the originator molecule. You have to run both side by side to show that they are exactly similar. Next slide, please. So these are some of the rules and regulations for all these tests. Because you're doing so many tests and the agency is making a decision on the quality of your product because of which is coming from the results of these tests. The conduct of those tests will go through a lot of scrutiny by the agencies. They will see that it has sufficient sensitivity to pick up all the impurities. If you're not, it has to be scientifically sound. You have to qualify it and show that it gives repeatable results, not that one time it gives, one time it doesn't. So you'll have to show some level of precision and accuracy. You also, and wherever possible, you will use orthogonal methods. If you're looking at glycans, you have 2 or 3 methods that can actually look at glycans. You will do all 2-3 methods to get the same result so that their orthogonality is measured. You will also give as consideration to the age of the batches, because whatever, when you do comparison, you have produced a product and there is something in the market which is already aged. Maybe it's already 1 year old or very close to end of shelf life, but your product is fresh and you are comparing it and saying we are equal. So that will also be scrutinized. Source of variance, so whatever variance you get, as I spoke earlier, whether it's coming from upstream, downstream, or is it coming from stability, or is it coming by the way you're handling your product, that source of variation also needs to be documented. As far as possible, if you're able to pick those variants and assess what is the impact, then that is considered to be with higher scientific rigor. Since we are doing these studies in campaigns, so all of this doesn't happen at one time, right? So the age of the entire development could be about 7 years to 8 years. So you're doing similarity in the first year or second year, then you're doing similarity in the fourth year. Even the people who are doing the test would have been, would have changed. So because you have different campaigns, and all of it is statistically analyzed, you need to have reference standards that are run in each of these campaigns so that you are able to bridge those studies. This is how it looks. This is our molecule, which is, I just downloaded this from the FDA site, the first biosimilar trastuzumab. This is how it looks. Each of those dots represent one batch. Okay, I have just put a few attributes here, which is high molecular weights and total fragments. So what you see here is, this is the biosimilar product. This is Herceptin sourced from the US region. This is Herceptin sourced from the EU region. So each of these dots is one batch. That means we have procured about 20 batches from EU, 20 batches from US, and these are our at scale batches. So these are, let's say, running in 6 scale batches. So each of that is a 6 scale batch. So basically you will show that multiple batches, you are able to control your product variant to this level of stringency. So if you see here, this is just 0.4, 0.5%. These are impurities, but which are very low. And then you have, this is potency, which is inhibition of cellular proliferation. If you see, the potency is close to 1, which means it is high, the potency is equivalent. And here again you see this is total fragments, which are about 2%. So this is the range, and this is coming from a statistical calculation, and hence all your batches have to fit within this statistical limits. So this is the level of scrutiny that goes for a therapeutic molecule. Now the fourth part of my discussion, which is differences. Now there are some differences in glycan analysis. Okay, once you have a difference, what you would do first is, you would go back to see whether you can control it in your process. If you are not able to control, then you will go back and look at the criticality of that attribute that is not matching. Now, for example, here you are seeing this is an attribute which is outside of the margin, but when I look at the function, the same batches have now come within the range. What it means is, there are some batches which are out, but you are not really seeing an impact on function. You will establish this completely to see whether it really doesn't have an impact. But sometimes you will have, patterns like this, where all your batches are out of range, right? So it means it is a real difference. There is a real difference. Now you will go back and look at what is this attribute, and is this attribute high criticality, moderate criticality, or low criticality. Now if this difference is very high in criticality, that means directly impacting, let's say, efficacy. That means your product will not be equivalent in your clinical trials. You cannot go ahead with such a product. If that attribute is not so critical or moderately critical, then you will look at ways to find scientific reasoning to support that difference. Can you go to the next slide? Okay, I am just giving you a case study over here so that you understand what these differences are. This is, I have just taken rheumatoid arthritis. So if you see here, this is how rheumatoid arthritis will look. How does rheumatoid arthritis occur? If there is overexpression of TNF, TNF is a molecule, protein again. tumor necrosis factor. If it is overexpressed, these are your diseased joints. It will show up in joints like this. And your healthy joints will have the optimum level of TNF. Now it means in the patient, TNF is produced more than that is needed. So in a biological therapeutic, there are two ways of action. One is, if something is not produced enough, then you can add it. For example, in diabetes, patients don't have insulin. So you are supplementing insulins, that is called agonist. And in cancer, you have cells dividing unwantedly. That means it is doing a function that you do not need. So you need to stop it. So one is positive, one is negative. So when it is an antagonist, what you would do is you would use these antibodies which will bind it and clear it. So the excess will get cleared. That's what happens here. So if you see, this is the antibody. It binds to soluble TNF and membrane bound TNF, both the forms. What is the job here? It has to just clear the soluble TNF. Once it clears the soluble TNF, you will be cured, right? Now what happens is, you have soluble TNF, you also have transmembrane TNF. That is, TNF is also found in a membrane bound form. It means the antibody will also bind this. This is the major function, it will remove this. But what happens is, as I earlier explained, because it has the FC region, it will also attract your immune cells. Once you have the immune cells coming into picture, the immune cells will kill this cell, okay, because of the cross-linking of the two cells, and this is not a desired activity, which is why when the glycans are different, what you would show is whether this activity is going to impact the major mechanism of action of my molecule. So you basically have to go a deep dive into the indications of the drug. So this is not a one size fits all kind of a approach. For every drug, you could end up with something very different, and then you have to do a very product specific assessment to see whether it qualifies the therapeutic requirements, and then you will design experiments to show that whether it is impactful or not impactful. Next slide. Oh, thank you so much. Got some water. Okay. So now this is, we found a difference in the glycan, right? So now we will do all kinds of experiments. I said you will do orthogonal techniques, right? So first is you are looking at the binding. So FC region will bind a FC gamma receptor. So if the glycan is different, it will show up in the FC gamma R3A receptor. So if you see here, these are US lots, these are EU lots, and these are biosimilar lots, and they are higher in binding, right? Clearly you are able to see in both forms of FC gamma R3A that there is a difference in binding, which means there is higher binding to FC gamma R3A. Then what we do is, just binding is a chemical reaction, right? Now we will go and see if I also supplement the macrophages, that is your PBMCs, how does the cross-linking happen? Does it really kill or does it have any activity? So now what we will do is, we are using the transmembrane expressing cells. Transmembrane expressing cells are also very transient, it is not continuous. So if you are overexpressing, that is also, you are artificially creating a system. And then you are running it with NK cells, you are running it with PBMCs. And this artificial system, you will change it into LPS stimulated monocytes. That is what actually happens in physiology, right? In all different ways. When you do it in all these different ways, which are physiologically more relevant, you see that the activity is coming inside and there is no activity here, right? So what we do here is, if you see, these are all the different cells that you can use. What is the sensitivity to FC glycans? The sensitivity is highest over here and lowest over here. However, biological relevance is lowest over here and highest over here. That means if you use a very super exaggerated lens, you will see it dirty. But if you apply the lens which is physiologically relevant, you will see that it is equal. So what we are supposed to do is, you need to show the difference, you need to understand the difference, you need to measure the difference with the super sensitive methods, and then justify it using biologically relevant methods, saying that I do understand there is physico-chemically there is a difference. However, that difference is not impactful in the patient. So it's an experimentally demonstrated justification, scientific justification. If you provide such detailed systematic justification, then agency will accept those differences. This kind of work is not needed in a generic because generic is an identical molecule, and that's why it takes so much time to produce biologics. Next slide. This is my final slide to show you what the entirety, what is confirmed similarity. So this is the entire process, the recap of what we just spoke. So you started with characterizing and fingerprinting the molecule, and then your cell line and process, and you establish biosimilarity. If it is not same, you went back. So it is iteratively you are going back, right? This is the first part. Second is once you establish a biosimilar candidate, you establish analytical similarity, you scale it up to manufacturing. Once you scale it up to manufacturing, again you establish comparative analytical similarity and take it to comparable safety and efficacy studies. Once this passes, then you do process validation, establish all your controls and submit it for filing and approval. And if you see here, because you don't do full clinical trials, your specifications of originator will always be wider and specification of biosimilar will be within that. Because you do not have the capability or the power of the full clinical. So you will always control your product within the originator specs. And that's your final analytical comparability. So analytical comparability is done in windows, in so many different windows. From that end to this end is almost about 7 to 8 years. Right? Next slide. That's the final. So that's the message.
Good evening, ma'am. First of all, thank you for the talk. I am currently doing my MSc in chemistry and I aspire to become a researcher in pharmaceutical firm. So whenever I bring this up, one of the point which people tell and discourage me is that in other, like other than this, if we take the organic synthesis and all which goes on in the pharmaceutical firm, the amount of chemicals exposure and the APIs and the excipients, it can have very bad health effects, mostly more to women also. So what are your thoughts on that? And then if you had faced any challenges, how did you overcome in pharmaceutical?
It's a very simple and straight answer. See, we have something called ESG in the company, environmental social governance. So environmental health hazard is part of the entire setup. You need to get clearance from pollution board first to set up the unit itself. We have something called certificate of establishment, certificate of operations. All waste management is controlled by local government authorities and everything has to be cleared for you to do it, and not just local government authorities. When we go for filing, there also we have to show how we are managing all the waste. So these are mandatory requirements. If you do not qualify it, you will not first of all get an approval to set up a company. And then even if you, let's say, somehow local, you manage to go beyond the regulations, you will still not be able to file your product. So this is kind of taken care. Yes, there are some discussions on when you get into the industrial area, you do, see or smell some fumes and things like that. They are purified and left into the air, which is why you have, a special economic zone which is full of only industrial units, right? So it does not have any impact on your health, but if you have a not so favorable, a smell, you should not be living nearby. That's all it is. That is why this area is called an industrial area. But it is not hazardous to your health for sure. I spoke about a very straightforward molecule. We also have something called anti-drug antibodies. So antibodies are protein molecules. They are not harmful to you. It is what is there inside your body is what you are making outside. But there are small molecules which are hazardous toxic molecules which are conjugated to the antibodies and used for cancer treatment. So it is like a missile. It goes in and then delivers where it has to deliver. Those come under hazardous chemical classification. So there is something called occupational exposure limit, OELs. So there is certified OELs, that is in this cubic centimeters of whatever volume of this space, how much of this molecule can be present and how much per day a person can inhale. There are clear limitations for all of this. You will not get approvals if you are not able to abide by that. And I can tell you for sure that nothing happens to you. It's safe.
Any other questions that we have?
Pretty new to the, I think, biopharma segment. So my question to you is, how much of robotics is in use currently? maybe in your organization or in general?
In general.
Yeah.
Yeah, so sample testing, so robotics is liquid handling systems we have, which is basically when you're doing samples, you add some volumes, you dispense volumes, then you move it, you wash it, you dispense. So that kind of robotics we have. We don't have robots walking around doing things. We don't have that. Okay, but they are all mechanized. Everything is mechanized. What I showed you, these are big bioreactors, and then you have sampling ports, and you have lines coming in, water lines. They are all, it has to be sterilized, right? The entire setup internally has to pass certain classification, the area classification and all that. So everything is controlled area, controlled rooms, and there are independent mechanical units. Bioreactor is a unit, chromatography unit is a unit, so all of this. There are manual intervention into this entire end-to-end process. It's not completely mechanized, which is what I spoke in my panel discussion, that if you are able to make it continuous processing, then it will be end-to-end, no manual intervention till you get the drug substance. We are not yet there, in India at least. There are some companies which are trying to do it in a smaller scale, but at a nascent stage now. So AI ML is a very common question that we hear nowadays. For discovery biology, it is very useful when you are looking for, when you're looking for leads, lead optimization, that's called discovery biology. When you, like for example, if you have an indication, you're looking for any molecule that will dock onto that particular protein or something like that. There is a lot of bioinformatics and hence all learning models and all of that, model predictions and all of that is in place. But in classical R&D, the work that we are doing, we want prediction from our own data, right? So for example, I said we have launched 8 molecules. It means we have a large repository of data and knowledge. And some of them is in print because we did not have digital. And last 4 or 5 years, it's all digital for us. But after digitalization comes the data analytics. So it's a stage. So first we have to make the data electronic in the sense that not everything is electronic. There's a lot of metadata which is handwritten, especially wherever manual interventions is there. Once everything is digitized, then there is a hope that we will reach a data analytics, and then these learning models will help us gain knowledge from our own experience. We are working towards it, but we are not yet completely there. But it will be very helpful to speed up conclusions and things like that, because the entire R&D is about 2 to 3 years. If we have ML, AI and things like that, it will reduce it further, is the expectation.
Anita, question. So, when with biologics, there are cases where someone has a reaction. So, is there a different way, say a blood test that can be administered instead of it being administered to a person before actually starting treatment?
You're talking about the thalidomide incident. So thalidomide was, infliximab is more injection site reactions. So,
you're talking of anaphylactic shock, is it? Huh? That's not thalidomide. That's an immediate
this is
reaction.
infliximab.
What?
Okay, we can discuss that. Okay.
Antibiotic.
MABs will, okay, so you should not, because it's proven in clinical trials, right? So it means it's a large population, we are looking at all. That's what. So it is, clinical trials are always statistically, it's powered, so you'll have 85% confidence interval and things like that. But deaths never occur, okay, in this. You can have adverse reactions, sometimes it is little more serious or less serious adverse, and injection site reactions are very common depending on the formulation. There are some citrate based formulations which lead to injection site reactions. So today everybody wants citrate free injection site reactions. That also is kind of, there are different schools of thoughts around it. We can discuss more around that. But there was one incident of thalidomide which was basically for avoiding morning sickness. That drug was given to pregnant mothers, and then the kids were born with fetal deformities and things like that. right? So post that only the clinical trials, the requirement of clinical trials and the stringency on clinical trials, all that went up after that. But all new molecules first go into animals. Now we are even trying to bring that down because for the sake of humans, you're killing animals. So that is where all your, predictions, and which is what I was speaking in the morning, how much of it can be brought down, because a large repository of data about protein structure, all of this is available, and hence it should be easier to predict such things. It's come a long way. It is much better than the early incidents, but there could still be injection site reactions and things like that. That's possible. But at least fatalities are not known.
Any further questions for Anita? Otherwise, thank you so much, Anita. A wealth of experience which you have shared with us. It's simply amazing to see how passionate you are about this business.
Thank you.
Thank you so much.